So far we've talked a lot about the cell, how the cell's organized and all kinds of stuff like that, membrane transport and a bunch of other stuff. But you know, we found one little thing. Yeah, what's inside the nucleus? We talk about the DNA and RNA, and that's what we need to talk about today. Okay, so let's just see where we go with DNA and RNA, and hopefully we'll teach you a little bit about this stuff. Okay, the music of life. That sounds really sweet, doesn't it? Okay, anyway. Let's go on. Well, first of all, let's get some definitions out of the way. First of all, DNA. DNA stands for deoxyribonucleic acid. So I get the DNA, okay? Deoxyribonucleic acid. And basically what DNA is, it's really the genetic cookbook. It has the recipe. Now people, if I ask them, I said, what does it have the recipe for? And they'll say, blue eyes, blonde hair, blah, 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 all that stuff like that. And basically, I'll take a pass, okay? What happens is DNA really has the recipe on how to make proteins. Proteins are important because, yeah, you know, the proteins in my eye might give my eye color and stuff like that. But what the proteins do is they guide all of our body processes. Everything needs a protein. Enzymes, all these things are proteins. And DNA has the code for all these body processes because these body processes are run by proteins. Okay, so that's called DNA. We'll talk a lot more about DNA in a couple of minutes. Okay, second definition we should talk about is RNA. RNA stands for ribonucleic acid. Well, where DNA has the cookbook, has the recipe, ribonucleic is how the proteins are made. Okay, what happens is that we get that code from DNA. Okay, what we do is we start this, we look at DNA and get the code for how to make a protein from DNA. Okay, so we get the code up in here in the cookbook. But what happens is when we make it because the RNA is what's going to make it for us. Okay, so we'll talk a little bit more about the RNA, and there are different types of RNA, which we'll talk about shortly. Nitrogen bases. Okay, nitrogen bases are really important. This is something that's good to understand. First of all, I have 4 + 1, which means that there are basically 4 nitrogen bases: adenine, thymine, cytosine, and guanine. I said +1 because what happens is those are according to DNA. So that's what we see in DNA. What happens in RNA, they actually substitute, and let's get rid of thymine, and they put in uracil. So they have uracil instead of thymine for RNA. Okay, so RNA has adenine, uracil, cytosine, and guanine. Okay, nucleotide is another definition. A nucleotide is one of these nitrogen bases: adenine, thymine, cytosine, guanine. A sugar, okay, and 1 to 3 phosphate groups. Okay, we'll talk a little bit more about that as time goes on. Now, this is a really important concept here, and it's fundamental. It's vital, and what we are called base pairs. Base pairs. And what base pairs are is they're a pair of complementary nitrogen bases. So let's go back to our nitrogen bases. Let's get thymine back in there because thymine deserves to be back in there in DNA. Let's talk about our complementary nitrogen bases. My nitrogen bases: adenine, thymine, cytosine, and guanine. What happens is in DNA we have 2 strands. One strand is one nitrogen base or nucleotide on one side, and on the other side, attached to the other strand, is another nucleotide or nitrogen base, and they're bonded together in the middle, okay, by hydrogen bonds. So what happens is we always know a couple things: that cytosine on one strand will always bond with guanine on the other strand. Adenine on one strand will always bind with thymine on the opposite strand. And the only thing that we'll have to talk about is uracil being substituted for thymine, but that's only in RNA. DNA does not have uracil, it has thymine. Okay, so cytosine will bond with guanine, adenine binds with thymine. So let me ask you another question: what binds with guanine? Well, cytosine, of course. What binds with thymine? Now, if you didn't say adenine, you're way behind the learning curve, so you better catch up. Okay, so that's what the base pairs are, and that's going to be important. And you're going to see about how that comes into play when we talk about DNA in just a minute here. A nucleic acid. A nucleic acid is basically a string of these nucleotides, okay, that contain the genetic code. So therefore, a nucleic acid is called, for lack of anything, DNA and RNA. Okay, so these are definitions. If you don't get these, maybe it might be a good idea to go back and review this one before you head on, because these definitions are going to be coming up time and time again as we continue with this discussion. Okay. Now let's talk a little about DNA. First of all, DNA, or deoxyribonucleic acid, was supposedly discovered by Watson and Crick. Oh, it's a red one. Probably should have put red, but Watson and Crick in 1953. And they actually have a book. I have a copy of a. Oh, they're probably out of print now, but they have a copy. They made a book, and the book is called The Double Helix, because basically what DNA is, it's like a double. It's like a staircase, like a spiral staircase. It's a double helix of a sugar backbone. They're paired with each other. And if I look over on the right side of the screen, you see two purple ribbons, and that's that sugar background, which is a ribose sugar. Okay, so the sugar is a ribose. Okay, so the sugar ribose double backbone, which is this purple thing that comes around here and spirals around. Okay, that's that. And what happens is between these two ribbons of ribose sugar, I have these nucleotides, those nitrogenous bases. Okay, nucleotides because it's a peptide, or the phosphate bonds and hydrogen and stuff like that. But what happens, I have these nucleic acids that are opposite each other, these nitrogenous bases. Okay, so what happens is, let's look at this picture. Let's just look at this. Let me get a smaller thing right here. So here's a thymine. What happens is that's on one strand. On the opposite strand, I have an adenine. If I look down here, here's a cytosine, and on the opposite strand, I have a guanine. Here's a thymine. I have an adenine, and let's see. I can't see the guanine down there. But anyway, you could assume that there's a guanine on this strand. The opposite side is going to be a cytosine. Okay. So this was first described, like I said, in 1953 by Watson and Crick. And you see, here's, the two guys up there looking very perplexed at this little model they have. The thing is, the fact of the matter was that the person that actually sort of pushed them into that was Rosalind Franklin, their lab assistant. The lab assistant said, hey guys, you know what? Maybe you should look at this. Starting to sound a little like drunk history, but it's not. Okay, you got to look at this. And actually, her work in structures, really, this double helix structure, actually enabled Watson and Crick to come up with this DNA type model that they talked about. So actually, a lot of credit goes to Rosalind Franklin. Okay, so anyway, that's that. So we have this sugar backbone, double backbone. Okay, two strands. Okay, and these nucleotides, the nucleotides are bound together by hydrogen bonds. So hydrogen bonds are holding this. We know those nitrogenous bases are adenine, which binds with thymine, guanine, which binds with cytosine. Adenine always pairs with thymine, cytosine always pairs with guanine. Remember that, remember that, remember that. Therefore, this is the beauty of DNA. If I know one strand of that, if I remove one of the sugars, one of the ribose sugars, okay, and the nitrogenous base that's attached to that, or the nucleotide that's attached to that, I know what the other side will be, that side that's removed. Why? Because adenine always pairs with thymine. So let's say if I cut this thing right down the middle of these hydrogen bonds right there, I know when I make another strand out here, if I make another strand out here like this, let me do it purple so that way we keep consistent here. So I have another strand that I'm going to make. What's going to be binding with the thymine? I'm going to have an adenine right there. What's going to be binding with the cytosine? I have a guanine right there. So basically, by knowing one strand, I know what the other strand is because it's a complementary pair. Okay. What happens is when we go back to one of our previous discussions, when we talked about cell division, we remember in our interphase, we got into our S phase, and the S phase is where what happens is now all the strands of DNA will split apart. Okay, there's an enzyme that will split that apart, and basically what happens by splitting apart, they could create a complementary strand on the other side because they always know that thymine is going to bond with an adenine, and cytosine is always going to bond with a guanine. Okay, and that's the beauty of this whole thing. So during cell division, each strand unwinds. So we have two separate strands. I'll have one strand out here. I'll have one strand out here. And basically, if this strand has let's say a guanine, I know that the strand on the opposite side, if I have a guanine, is going to be a cytosine over there. Okay, and if I have a strand over here that's an adenine, okay, when they create that other strand, what's going to happen? That other strand up there is going to be a thymine. Okay, so basically they could actually get these things really paired well. So from one double strand that will split, I could create 2 strands from that single strand, and that's the beauty of it. And it's quite simple. All you got to know is which order those nucleic acids, those nucleotides are in along that ribose sugar strand. Okay, hopefully that makes sense to everybody. It's really pretty basic and pretty simple when you think about it. Now, sometimes when this happens, there might be a mistake, and when we get a mistake, that's what we call a mutation. Okay, and we'll talk a little bit more about mutations down the road a little bit. Okay, well, again, when Watson and Crick started looking at this double helix, they first discovered the nucleus of cells. Okay, that's where they are. Okay, and the DNA and the DNA stays in the nucleus, not the RNA. The RNA may leave the nucleus, and we'll talk about that a little bit later on. Okay. We know that it's an organic molecule. Why? Because it's organic. Because it's carbon, hydrogen, and oxygen, which I know is organic. Also some nitrogen and phosphorus and stuff like that. So basically, that DNA is that inherited genetic material. We have the code to make all those proteins, and all those specific traits that may be dictated by a specific protein. Okay, are on that in the chromosome in a certain area called genes. Okay, so each gene is really a segment of DNA. So if I have this long strand of DNA, periodically along the strand there'll be a certain segment with an appropriate order of these nucleic acids along that strand, and that area right there that has a specific code for a specific protein, that area would be called a gene. Now, interesting thing enough, when they started this genome project, where they started to map the chromosomes, what they recognized was there's a lot of material on my chromosomes that actually don't mean anything. Okay, they're empty. They have a worthless code. Some people think that they might be old viruses that are now no longer there or extinct, because the way a virus works, and this is probably very appropriate in the COVID-19 thing, is that what happens is the viruses are either pieces of DNA or RNA, and they're encapsulated by a protein coat around the outside. And what they do is they latch onto the surface of a cell, and when they latch onto the surface of the cell sort of like that endocytosis wraps around them. Okay, remember we talked about that earlier? So now it's all coming back. I like things will come back down the road. The cell membrane wraps around them and takes them inside the cell. Or where do they take them to? They take them to a lysosome. When they take this virus, the spiral particle, which is again either a DNA or an RNA. If it's an RNA virus, it's called a retrovirus. Okay, but they take this DNA particle that's still in this capsule. It's protected. It takes it to the lysosome. What do the lysosomes do? They have acids and other types of enzymes. They destroy the protein. And as they destroy the protein, they release the virus. The virus gets out, gets into the area of the nucleus, and incorporates itself in its own DNA and starts to make more viral particles. It keeps on duplicating itself, because what does the DNA or the RNA do? Has a tendency to duplicate proteins. So it duplicates more of itself by its genetic code in that segment of DNA or RNA that specifies a particular virus. That's how viruses work. That's why we can't kill viruses. Viruses were never alive to start out with, okay, to begin. And what happens is once they get incorporated in our body, guess what? How are you going to get rid of them? Because they're in a bunch of cells. And you can't get rid of these little guys. They're there forever. So we could sometimes help to control them, okay? And now when they're talking about RNA vaccines, the idea behind RNA vaccine is to be able to take the genetic code of part of that virus. And they're talking about, like with the COVID-19, the protein code on the outside. They can know what that protein, the code would be for that particular protein, because that code is in that RNA that's in that virus that's able to make that protein. Okay? If they take that protein code and make an artificial piece of RNA, okay, Um, and what they do is then they inject that in the body some way, or put it in a cell or something like that, and install it in the body. What happens is then the body's immune system will recognize that being abnormal and will create an immune response. So therefore, when you get infected by the full virus, as compared to just a small little chunk of what the code would be for that protein, because now the body recognizes not the virus, but it recognizes the protein on the outer surface of the virus, it'll destroy it before it could do any damage. And that's what they're talking about with these RNA vaccines, okay? So what do these genes do? Again, they have all that genetic information, and the genetic information determines traits by controlling protein synthesis and regulates body activities. So yes, in some ways it does tell us our eye color, stuff like that, because those are dictated by the protein that makes that particular that particular color. RNA. I said DNA has the inherited genetic material, has the code. RNA relays the instructions from the genes that helps to guide the synthesis of proteins and amino acids. Amino acids are going to be proteins. The difference also is where DNA is a double strand, RNA is a single strand. Okay, and that's another large difference between the two. And I'll talk a little bit more about that. I love this picture probably because I was a Game of Thrones fan, and you know, everybody was a Daenerys fan until the very end, maybe. But you'll get the implication here. Okay, DNA, RNA, single strand, double strand. Okay, so that's a little. I like that picture. It's sort of cool. Okay, so now we know what DNA is. And DNA is what makes up our chromosomes. I've mentioned chromosomes a couple times. And what chromosome is, it's basically a long molecule of DNA that's coiled. It's held together by various proteins. So there are various proteins inside that hold this coil. See up here in the top of the picture up in here, if you look up in here, when we look at this little diagram on the top, you see our DNA sitting up in here. What happens, these proteins sort of coil it up. So now all of a sudden, and that coil it starts to coil up. It looks like a big X of ramen, you know, all coiled up in there. And that's just all the DNA. It's sort of smushed together in this X type of a pattern. Okay, X like this, X type of a pattern. Okay, we have 43 pairs of chromosomes. Okay, 43 pairs. Okay, everybody does. Now, I can't say everybody because sometimes there are certain genetic errors where we might have too many, or sometimes too few. Typical example is Down syndrome. In Down syndrome, the 21st chromosome, instead of having 2, has 3. It's a 21. It's called a 21 trisomy. Okay, but we have 23 pairs of chromosomes. So overall, in each nucleus of our body, we have 46 chromosomes, but they're in pairs, so 23 pairs. The 23rd pair is called the sex chromosome. And the females, it's an XX, and the males, it's an XY. Okay, so XY in males. And the thing that's interesting is on the Y chromosome, there are certain types of genes that are only on the Y chromosome. Therefore, there are diseases that only males can get. A typical one, I think I had probably mentioned in an earlier discussion, was Duchenne's muscular dystrophy. There's different forms of muscular dystrophy. So females can get muscular dystrophy, but not Duchenne's muscular dystrophy, which is the most severe type. And the problem is a code for one of the proteins in muscle is on the Y chromosome. And therefore it's only going to be in males because males are going to be XY. It's only in the Y chromosome, and therefore it makes an abnormal protein, which results in the muscle weakness associated with muscular dystrophy. That's why, you know, they try to find a cure, but you know, the only way you're going to find a cure is being either something that's going to be able to change that gene on that Y chromosome, but that's going to be sort of difficult to do. Genes, again, are the hereditary units. They're aligned along chromosomes. So there might be, if you looked up in this picture, there might be a gene here. There might be something here, here. I mean, there are many. There could be up to, you know, thousands of genes on an individual chromosome. Okay, and so some of the genes that they have are only a couple sequences long. Depends upon how many amino acids are in that protein. If it's a very small polypeptide, or a small peptide, which only has a couple amino acids, the gene unit is very small on chromosomes. However, many proteins are just monstrous molecules, and these monstrous molecules will have a gene that's really, really long on that chromosomal strand. Okay, simply because you'll explain what it is. We'll talk about codons in just a minute. Okay, so these genes are variable sized units. Some are big, some of them are small and very short. And again, this is where we have the code. Now, if we looked at the nucleus of most cells almost all the time, through the majority of the time the cell's sitting there, basically, guess what you're going to see? Little X's? No, you don't. What you see is this clumpy, lumpy, bumpy, granular appearance of the nucleus. And this is called chromatin. Okay. Now we talked about this before, that what happens is for the majority of the time, with the exception of a couple of very short periods of time during mitosis, the chromosomes are basically long strands. They're all stretched out. They don't assume this X-shaped or what we call a dyad structure, but they're just all strands. Now, it's not like they're haphazard, they can go anywhere they want. They're in a certain territory or unit in the area of the nucleus. But what happens is it gives it the nucleus that granular appearance, and then not that X shape that we normally associate a chromosome with. And the reason why it does that is by taking and unwinding this X and just having a long strand of that DNA material, where it allows better access to the gene areas, so they're not really covered up, or they're not hiding in all those little nooks and crannies that we see in our ROM and X up over there. Okay, so that chromatin is that diffuse granular mass that we see in the nucleus, which is almost all the time, except for a very short period of time during mitosis, we see that granular mass. We don't see the typical chromosomal appearance. Okay, one other word you probably should know is genome, and genome is basically the total genetic information in a cell. Okay, so in other words, each cell has the same genome, unless there's been some type of a mutation or a change in that particular cell. But the genome is basically the total genetic information in the cell, which means that cell has the genetic information to do everything that the body wants it to do. Pretty cool. And that's the nice thing about DNA. Okay, those are my skinny genes. Okay, well, that's sort of a different type of a gene, I think. Okay, now again, only during a certain portion of mitosis does the DNA actually condense into what we call a dyad structure, which means it's two sides. Okay, and each of those is called a sister chromatid. So I have a sister chromatid here, sister chromatid here, and basically they're genetically identical. They're identical. So when they come together, they're genetically identical in this X shape. So what you see over here is what you see over here. Same thing. Okay. And the reason why is because they came from the same original chromosome. The same original chromosome in replication created two sister chromatids, and they finally come together. They're joined together by little in the center, which is called that centromere, which we talked about how the protein actually attaches to that. So when the spindles start to spread and go towards each end of the pole of a cell during division, what will happen is it pulls the sister chromatids apart right at that area of the centromere. Okay, and we talked about that in our previous discussion. Okay, if you don't remember that, go back and look at that one. You'll see what that's all about. Okay, now this is sort of an interesting thing that they found when they did the genome. They did what's called the Genome Project. And the Genome Project was sitting down to map out all the DNA, all the chromosomal material that we have in our body, to know where all these genes are. Okay, it was pretty successful. They did a lot of that. So what happens is, again, each chromosome is one long molecule of DNA. Okay, and each DNA has regions. Okay, along that. And those things are called genes. So when they did this Genome Project, what they wanted to do is they wanted to provide an address for each of those genes, so they know where each of those genes would be located. Okay, along that entire length of DNA. In which chromosome. Okay, now this was a monumental project, because again, each DNA molecule may have a ton of genes. Okay, so basically it was a pretty daunting type of a thing. Okay, and each gene has the information that's needed to synthesize a particular polypeptide or protein. Polypeptide is a protein. Peptide is two or more amino acids that are put together. A polypeptide is multiple amino acids, and then basically that polypeptide is actually a protein, because a protein is a long string of amino acids. They're folded and clumped and all kinds of stuff like that. So this gene can be used as either as a direct process, okay, such as a protein to do something or a particular trait that we might exhibit in the body. So the most correct answer is, what do genes tell you about? They tell you how to make a protein. They tell you how to make a protein. And that protein might be responsible in driving an individual process, body process that we need for us to be able to live, or it might be important in creating some characteristic which makes me look different or be a little bit different from everybody else. Okay. Now again, errors in gene result in things like disease or an abnormal function of cell. And they're actually talking, and this is this isn't new, this is actually old, realizing that what happens is that genes people talk about the genetic basis of cancers. If there's some type of an abnormal cell division, we have a mutation in a particular gene, and that what happens is that causes maybe more replications and all kinds of things that results in cancer. They're called oncogenes and stuff like that. And we won't get into that because we don't have the time to go into a lot of the clinical aspects, but my feeling is that what's going to happen is down the road, when we start to look at treatments for cancer, is either going to be modifying the immune system so we're able to recognize these things earlier, or something that we're able to do something with the gene that's going to change that. Now, what happens is they were able to identify and provide addresses by 5 different characteristics. Okay, first was which chromosome is the gene on. So that was the first thing. First idea, we have 23 pairs. Which of those chromosomes is a specific gene on? Second is which arm. When we look at the arm, one part of the chromosome is a short arm. The other part of the chromosome is a longer arm. So is it on the short arm, and they designated that as a P? Is it the long arm? They designated that as a Q. Okay, so which arm? Then they divided the arms into what are called regions. Okay, so they look at regions, okay, and say that this, so here's my long arm. This is chromosome 7. Okay, this is the long arm of chromosome 7. Okay, here would be the centromere, would be right in here. Okay, so this is the long arm, the longer of the two. That's the long arm. Then they divide it into regions. They put the region in right here. So this is region 3. Okay, so this would be region 3. Then they looked at that region and says, you know what, we have to be a little more specific, and they broke it down into what are called bands. So they look at it and say, okay, let's take this band right here, and that's called a band. And they said, well, we have to be a little bit more specific, so some of these genes are relatively small. Then they went to subbands, and that little area right there is a subband. So what they were able to do is they were able to, if this little area right here, where I'm going to put my purple line right there, that little area would be described as chromosomal location as 7, 7th chromosome, Q, long arm, 3, region 3, band 1, and subband of that 0.2. Okay, because each subband was a point something, 0.1, 0.2, stuff like that. So this area, the purple area, would be designated as 7Q31.2 in regards to where that particular gene is. Okay, now what's the significance of that? Let me show you that. Well, let's look at this. Let me get rid of this right here. And this is the 7th chromosome. Obviously, we see that this is chromosome 7, 7th chromosome. Here's my long arm, so the 7Q. Okay, look for region 3, and region 3 is down in here. Okay, and then band 1. Okay, band 1 and 0.2, and this area right here. Okay, which would be designated as 7Q31.2, would have the gene that would actually be responsible for producing cystic fibrosis. So in other words, if that gene is wrong, if that gene is okay, it's fine. But if that gene is mutated, or if there's a bad gene at 7Q31.2, the resultant disease would be cystic fibrosis. So that's where this became important. They were able to look at the particular area on a particular chromosome, okay, by chromosome, by arm, by region, by band, by subband, as to be able to determine where a particular genetic problem would be. Now they know exactly where to go if they ever should get to the point where they can do something about it. Okay, so that's all about that. And to be truthful with you, in cystic fibrosis, in the past, it seemed to be that, you know, I mean, there wasn't really much of a treatment because it's a genetic problem. However, more recently, they're finding some changes and some things that they're able to use that may have some progress. So maybe within, in the next few years, you can hear some nice things about cystic fibrosis coming up. At least I hope so, because cystic fibrosis in the past was a highly lethal condition, usually causing death, usually in the 20s, because the lungs get trashed, as well as the GI tract gets trashed as well. Because what happens is a problem in regards to how the body handles sodium, and as a result, where sodium goes, water always goes. Remember that. Where sodium goes, water goes. So the problem is all these secretions in the lungs and in certain ducts and some glands in the body are so thick that they get blocked up. And as a result, it trashes the structures, and eventually the lungs will fail. And when the lungs fail, because there's so many repeated infections, all kinds of mucus and garbage in the lungs, they'll end up dying. Okay, so hopefully, hopefully this new stuff that's coming out in regards to cystic fibrosis will be helpful. Okay, now DNA replication. Now, we talked a little bit about this before. What happens is during that S phase, okay, when we go to that S phase during interphase, remember what's going to happen is the nucleus has how many chromosomes? 23 pairs, okay, even though they're not in that dyad structure. What happens is now I just have these long strands, those the long strands like we talked about that we mentioned before, okay. What happens is now during that S phase, I want to take those 23 pairs or 46 chromosomes and I want to duplicate them. So I want to double it. I want to double it, okay? And what they do, and this is again very simple, okay, that they could make an exact duplicate of each of those strands by actually what they do is they split and they unzip the strand right down the middle. Okay, there's a particular enzyme which I'll show you in a couple of minutes, okay, which actually takes and unzips the strand of DNA right down the middle, right down the middle. It breaks the hydrogen bonds between those, here, here, all those little dots in there. Those are little hydrogen bonds. It breaks those hydrogen bonds, okay, and it unzips it. So what happens is the two strands become one coiled strand. But I know on that one coiled strand, I still have those nitrogenous bases. And if I have a nitrogenous base that sits out there, such as a thymine, I could actually build another layer of sugar on the other side, and if I have a thymine, I know what's going to happen is I'm going to grab an adenine to go on that other side, put some bonds in the middle. Now all of a sudden, by doing that for each base pair all the way down that, I've created a complete double strand out of one strand, and that other strand created its own double strand. So that it's actually able to create two strands out of one by unzipping down the middle, matching the base pairs from one side to the other, creating another sugar backbone, and as a result, what happens is now one becomes two. And that's what happens in that S phase during my cell life, okay? So it breaks down those hydrogen bonds, it unzips it, and those nucleotides are drawn to the opposing nucleotide. Again, adenine always looks for thymine, and cytosine always looks for guanine. And basically, what happens? Another enzyme called, DNA polymerase. We'll talk about that in a second. Actually, sort of bonds everything together, gets everything together. It's like the super glue of DNA. And as a result, now I have two DNA molecules with the identical sequential arrangement of nucleotides. Okay, where if I had one strand over here, which has my adenine, the other strand that would slip out over here, let's just look at this, okay? So we have this unzip over here, and let's look. So here's let me use a smaller thing right here. Here's a thymine, and here's an adenine. So when they make that other sugar band over here to make that double strand, what's going to go here? A thymine is going to go there. What's going to go there? And adenine is going to go there. Okay, so we're going to find it. We're going to put an adenine there. Whoops, let me get it a little bit bigger. I'm going to put an adenine there, and I'm going to put a thymine there, and now all of a sudden, now they're able to build that second strand. Okay, completely. Very neat. It's you think it's so complicated, but it's actually so simplistic and beautiful in the way it's made. by this DNA replication. And now, this is for your own information. I wouldn't spend a whole lot of time looking at these, but there are basically about 6 enzymes that are involved in this. And again, don't remember these. Maybe there are a couple that I think that are really important. Most of them aren't, okay? If you have to remember, if you really want to impress your neighbors and friends and never have them ask you out for dinner again, probably what I would remember is I remember helicase, because what helicase is actually the important one. That's what unzips the double strand of DNA. Okay, it breaks those hydrogen bonds to create 2 separate ribbons, or a template for forming 2 complementary pairs. Okay, so that unzips the double helix into 2 strands, and those 2 individual strands then make their own double helix by binding complementary nitrogenous bases. Okay, the other ones I think are important are the DNA polymerase. Okay, and basically this is it sort of like puts it together. It's that super glue that holds the things together. The other ones are okay, a gyrase. Think about gyration, it's twisting. That DNA is twisted. This sort of opens it up and unstrands it and stretches out a little bit, so it's easier for those base pairs to line up and stuff like that. And primase just sort of sets like a little bit of glue or cement, so that what happens, those nitrogenous bases could be matched a little bit better. And ligase basically fills in the gaps when it's all done. That's all it is. So that's for just for your information, for the fun of it. Now again, this is just what we're talking about with DNA replication. And you can see on the left how the helicase has unzipped these strands, and what happens is that from that single strand, or that double helix, I have 2 single-stranded twists. Okay, and then what happens is then the base pair will bind. Okay, you actually see where this cytosine broke away from that guanine from that cytosine, stuff like that. And all you need to do is, when it splits, now what happens is, here's a thymine that's there, an adenine, when they build that sugar, is going to bond to it. Here's an adenine, I know a thymine is going to bond to it. Makes sense. And this also shows the same thing on the right hand of the screen. You have the chromosome broken down into DNA, okay, that we'll see. And here's the helicase that unsplits it down the middle. And what happens is I create 2 strands from one. And again, it's adenine's always going to look for thymine, and cytosine's always going to look for guanine. And that's what happens. Now, you don't have to remember this, but I think it's it's also another little cool thing. What happens is each strand does not produce the same. I don't know why. Maybe they just figured that, hey, you know what? Whoever designed this system years and millions of years ago, probably says, you know what? We got to really screw with the brains of all these A&P students down the road, and tell them that, guess what? One strand doesn't duplicate the same. What they have is what's called a leading strand. And a leading strand, basically, as it unwinds, it builds it from the bottom up. As it unwinds, once it unwinds and once it splits, unzips, then what happens is it builds from this way up. Okay, what happens is then there's what's called the lagging strand, and that's the opposite strand, and it goes this way. It builds from this way down. Why? I don't know. Again, just to play with our brains. I guess that's what it is, but you don't have to remember that. But anyway, just remember that the double strand is unzipped right down the middle, and therefore, because if I know that adenine always binds with thymine and cytosine always binds with guanine, I could duplicate a second sugar backbone with the appropriate nucleic acid or nucleotide base pairs, and it works really well. Okay, and that's what happens with that. Now, that's a little about DNA. Now we get into that other thing called RNA. We know DNA has the code, okay? But what do we do with that code? We know it now, it replicates in that S phase. So now what happens is, when that cell divides, like we talked about in the last video, I could actually have 23 chromosomes, 23 pairs of chromosomes that will go to each part of that cell. And when it cleaves down the middle, now I have 2 cells still with 23 pairs of chromosomes. But we have what's called gene expression. And gene expression is, even though the DNA has that code, and I've replicated the DNA so that when the cell divides, I could have an equal DNA on both sides, a normal DNA, the same as the parent. In other words, the parent started out with 23 pairs, and each of the progeny will have 23 pairs of chromosomes. Okay, big deal. Okay, I got to still run body processes. And how do those body processes run? They run by RNA. Okay, and RNA is the workhorse of gene expression. Okay, genes are on the DNA, but the RNA is the workhorse. Okay, now RNA stands for ribonucleic acid, ribonucleic acid. Okay, RNA, and basically it's a ribose sugar, but only a single strand, like we showed in Daenerys, or before from Game of Thrones. Okay, and ribose is basically a 5-carbon ringed sugar. It's the backbone. Okay, has phosphate, just like the DNA did, and it has nitrogenous bases. Well, almost like the DNA. Okay, it still has cytosine, guanine, and adenine, but instead of thymine, it has something else called uracil. So there's no thymine in RNA. Uracil takes the place of thymine. Why? Probably the same guy says, you know what? Let's just put something else in there just for the heck of it. Okay, and what happens, purines and pyrimidines are two types of proteins. Purines, adenine, and guanine are purines, pyrimidines, cytosine, uracil. Don't worry about that. That's not important. Okay, so this is the still single, always. Okay, from RNA, always a single strand. Okay, and that's going to be important. Well, the plot thickens. We can't stop there. That's just too simple. What happens? There are 3 different types of RNA. 3 different types. One's called messenger RNA, okay? And messenger RNA transcribes the code off the DNA. The DNA has the code, and the messenger RNA transcribes the code. We also have what's called ribosomal RNA. Well, guess where the ribosomal RNA is? Well, it's a site of protein synthesis, and where does protein synthesis occur? In the ribosomes. You should know that. Okay, so ribosomal RNA is basically the factory where the protein is going to be made in the ribosome. Okay, so again, we talked about how the ribosomes are basically 2 subunits, one with ribosomal RNA and the other one with another protein just to hold together. The third type of RNA I have is called transfer RNA, and basically this translates the code and brings the appropriate amino acid to put in the mix. I'll show you how that happens in a little bit. Okay, so each have a specific role to carry out from instructions of how to make a protein. Let's talk about that. First of all, let's talk about messenger RNA. Messenger RNA is manufactured in the nucleus and then migrates into the cytoplasm. Okay, it's again, it's like all of the RNA. It's a single strand, sugar phosphate background, the ribose background with phosphate. Okay, has many exposed nitrogenous bases, which are in triplets. Now, this is another word that you need to know. Codon. Codon is what the definition of a codon is 3 consecutive nitrogenous bases. It might be adenine, cytosine, guanine, cytosine, guanine, cytosine, adenine, uracil, cytosine. It's 3 individual nitrogenous bases, and that's called a codon. Now, what does that codon mean? Each amino acid has a special codon name. So there's a codon for each specific amino acid. Codon for each specific amino acid. There's also what's called a start codon and a stop codon, and that's going to be a little apparent a little bit, but probably more apparent in our next discussion. Okay. So what it does is it transcribes the code from DNA, so it can be taken to the ribosome and the ribosomal RNA for protein synthesis. It's the code for the amino acid synthesis, amino acid sequence. So it sort of acts like a memo, a signal or a memo to the ribosome that dictates the sequence of amino acids used in creating a protein. So where is the code for making that protein? Well, the DNA. Okay, we all know that. That's a piece of cake. Okay, so let me just draw it here. Let me just give you an idea. I'm not going to draw it, squirrely, because then it gets in the way. So here's the strand. Here's the sugar, the ribose sugar. Okay, and what happens if it gets unzipped? Okay, let's take. I can't remember what the colors were. Okay, let's say this is an adenine. Let's say. Don't go to colors as before. Let's say this is a thymine, and let's say this is a cytosine, and this is a guanine. Okay, so when these amino acids are exposed on DNA, what happens is RNA builds another sugar backbone. And what it does is, if that is, I said that was adenine, right? If that was an adenine, it normally in DNA would bind to thymine, but in RNA, there is no thymine. So it's going to bind to uracil. Here's my thymine on the DNA, which is going to bind with, or it's going to attract adenine. Here's my cytosine, which is going to attract guanine, and here's my guanine, which is going to attract cytosine, if that was the way it was. So what happens is now that when the DNA splits, the code is going to be read. Now, I mentioned a codon is 3. So let's say these 3 amino acids. Let me do it different. Let me just do it here. These three nucleic acids are a code for a specific amino acid, which is this. Basically, this code is a code for amino acid, and this is the complementary base pairs with the substitution of uracil instead of thymine for that amino acid. So now this messenger RNA knows what this amino acid is on DNA because it has the complementary base pairs, the opposite. Perfect. Okay, so it transcribes the code. So DNA has the code. That code is transcribed on messenger RNA by coding with the opposite, okay, or nitrogenous base. I'm done telling you, Carol. I'm done being this guy's messenger boy. He can leave the nucleus and do it himself for all I care. Yeah, okay. That's what the DNA and the RNA at work. Okay. Anyway, let's go. So now we know that the code is read off the DNA by the messenger RNA. What happens is I need something else to make that protein. And what else I need to make that protein is ribosomal RNA. Okay. Now ribosomal RNA is manufactured in the nucleolus. Where are ribosomes manufactured? In the nucleolus. And then ribosomes, we know, are ribosomal RNA as well as protein that holds it together. But what happens is after they manufacture them with the ribosomes, they migrate to the cytoplasm. Where do they go? Attach to the endoplasmic reticulum, hence the rough endoplasmic reticulum. Now it's making sense. Also, some will go to mitochondria and stuff like that because we know we have mitochondrial rRNA in there. Again, 2 subunits, large and small, contain 60% of the rRNA and about 40% protein. That's not work. But what happens is rRNA is the workbench or the site of the protein manufacturing in the cell. So that messenger RNA is going to do what? It's going to go to my rRNA in the ribosome. It's going to take that code that it got from DNA by matching the opposite nitrogenous base pair on that single strand RNA. It's going to take that single strand to the ribosomes, and the ribosomal RNA is going to take it and start to decode it. Now comes in my third factor, and this is what we call tRNA, or transfer RNA. So now along my rRNA, I have my messenger RNA with a specific codon, which is those 3 nitrogenous bases. They're sticking up. So this transfer RNA, which is manufactured in the nucleus and then migrates into the cytoplasm, is basically a single strand. Now, where I looked at my messenger RNA is basically a strand. My ribosomal RNA is more nodular. Okay. My transfer RNA looks like a 4-leaf clover. Is it like this? Come up like that. And on the top of that 4-leaf clover is attached an amino acid. Now, which amino acid is attached there? Look at this down here. Now, this is the code. This is what's called the anticodon. You remember that what happens was that the code for a specific amino acid on the DNA, let's say the code for a specific amino acid was, let's say we have a thymine, a cytosine, and a guanine. Okay, so that was on my DNA. So it's DNA here. My messenger RNA came by, looked at the DNA that was exposed because it unzips, okay? See, thymine, cytosine, guanine. What the messenger RNA is going to make is going to make a strand that's going to be adenine, guanine, cytosine. And this right here is called the codon. Got it? Because thymine always binds with adenine, cytosine always binds with guanine, and guanine always binds with cytosine. Now, if this was adenine, this would be what? That would be uracil, because there's no thymine in RNA. That messenger RNA then goes to the rRNA, lays it down on a bench, okay? And here's my adenine, here's my guanine, here's my cytosine sticking up. Now my transfer RNA, on the bottom of it right here, has what's called the anticodon. So if that was the anticodon, what you'd see is the bottom of it here to match what would bond, what would attract an adenine? Uracil. What would attract a guanine? Cytosine. What would attract a cytosine? A guanine. So at the bottom of here, you'd have this anticodon with a uracil, cytosine, guanine. Okay, that would match that. And on the top of that would be the appropriate amino acid for this particular code. Wow, that's beautiful. I mean, simplistic. Threes. Everything comes in threes. Okay, it's actually great. Here was the code. I transcribe it with the codon in messenger RNA that goes to my ribosomal RNA that lines it up and then orders the transfer RNA to bring in the right amino acid based upon this codon that would match an anticodon up there. Now let's just go up here to this. Let's go up to where we started. Let's look at this. Okay, so this is right. This is that's a transfer RNA for arginine. Okay, and there's a number of different codons that we'd be looking at, but I don't want you to look at that. In fact, let me just erase that so you don't see it. What I want you to do now is I want you to think, what would be the codon in messenger RNA for arginine? Did I hear that? The codon, cytosine, guanine, cytosine. So let's look over here. Is that there? Oh, right there. There it is. There's that little puppy. So that's one of the appropriate cytosine, guanine, cytosine. And it brings the right amino acid to there. So we have the anticodon on one end of the transfer RNA. We have the amino acid on the other. So what happens is the DNA had the code. Messenger RNA read the code by getting the apposite nitrogenous base, takes it to the ribosomal RNA. Ribosomal RNA looks at those codons, which is a triplet, okay, and orders the right transfer RNA to bring the appropriate amino acid to the table. And then what happens is when it does and brings down there, it binds, it reads it, and what happens is now that transfer RNA, or that messenger RNA is a long strand, and it'll read it codon by codon by codon by codon, as long as the protein numbers of amino acids would be there. And again, that's why there's a start codon, because there'll be a codon to say start, and there'll be a codon to say stop, which is basically some set of 3 nitrogenous bases. And when it gets to the start, it starts reading it. The ribosomal RNA tells the transfer RNA to bring the right amino acid to there, binds, the anticodon matches with the codon, which is a complementary nitrogenous base, leaves off the amino acid, which then binds the next one, which 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 binds the next one, and I have a string of amino acids that's called a polypeptide, which is a protein. That's how proteins are made. That's how DNA and RNA do it. DNA has the code. RNA is the workhorse, is the manufacturer of it all. I love it. It's really pretty neat the way that happens. I'm starting to get a little teary right here. I mean, if you can see, yeah, I'm starting to get a little shaky and jittery. I'm starting to goosebumps all over. That's the way it works. It's very simple. Now, if you didn't get this now, we're going to talk more about this, and we'll go over it again in the next video after this. But this gives you a little bit of idea, so the next video you can sit back and relax a little bit more and get it. What happens is I usually find it takes about 3 or 4 times to hear it until somebody says, "Oh, why didn't I get that the first time?" I mean, that's pretty simple. Okay, so basically that's how we make a protein. So messenger RNA reads the code off the DNA and takes the order to the rRNA in the ribosome. The rRNA is the place where the protein is going to be formed. The code off the messenger RNA, which came from DNA, is interpreted by the rRNA in codons on the messenger RNA, which is triplets, 3 pairs of nitrogenous bases. Orders the tRNA then to bring the correct amino acid based upon the tRNA anticodon, which is again the opposite nitrogenous basis from the codon. Okay, and then what happens is it brings the appropriate amino acid, which is represented by the anticodon. Okay, which is then previously represented by the codon, which is previously represented by the triplet on the DNA. Okay, it brings the amino acid. Put those amino acids together. I have a polypeptide. Polypeptides are a protein. Proteins formed. That's how DNA does it. It's great. I love it. Okay. So hopefully this gives you a little bit of an idea as to how DNA works. We've solved the mystery of life here in the last 56 minutes and 53 seconds. We know how it happens now. We know that mystery of DNA, and again, the DNA is different from person to person. It's a genetic inherited qualities from each of our parents. And while DNA is a really complex, long, twisted, convoluted structure, its function is really pretty simple. And that's when you break it down like that. How the simplicity of it all, you say that is really pretty neat. Someone who designed that was really thinking outside the box and did it right. Okay, and that's where I'm going to leave you today. Next time we're going to go over this again in regards to that process, maybe a little bit deeper next time, but hopefully this lays enough groundwork so when we see this again in my next video, it'll make a lot of sense to you. Okay, for the meantime, stay safe, be good, and we'll talk to you soon.